Welcome to the Summer Academy 2020 at Kaiser Neuromap Institute. We're going to be talking about aging and EEG and our response with the default network training to aging and related disorders associated with increasing age. All right, as I talked in our last video about how we look at EEG, one of the elements of EEG is how we increase our brain rhythms each year as we grow through childhood into adulthood. And this is going to be on the other side of life where we start to slow down. So I talked about this in the Unity years video, how we're increasing our sensory sampling rate, overall information transfer rate each year, up to a point. Then eventually we start to reverse that pattern. All right, so we're going to do, typically we record 19 channels on the head to get an assessment of what's going on with individuals, find out where to do the training, to do the operant conditioning to improve behavior. And I just want to bring this one up again, just because I found this very nice slide to talk about this. You know, it's always interesting when I see these papers out there where someone's done this amazing analysis on the surface electrodes. That for me is like doing an amazing analysis outside of a ballpark of what's happening inside the ballpark. You got to know who the players are. You got to know what's going on inside, and it's amazing. I in fact recently was looking at this immense amount of work, but it's using the referential channels. Well, the referential channels are composites, and there's a nice slide that they created. And this is it, just to show how, for instance, you have two sources of signals and how they're going to contribute to the electrode coming in that's recording them. And you got these composites, and that's why you have to do the subtraction, that's the Laplacian, to get a better clear where the unique voltage contributions are. And then you triangulate it down to sources. And this is just a version of the Brodmann Montage I created in 2006. This is the regional view, and then you go down even to the areas. All right. Okay, so we take the Laplacian signal, or usually we go down to the Brodmann Montage signals, and we figure out what's the activity level, and then what's the shared activity level between sites. And from that, we can get EEG coefficients that are meaningful. They're associated with the habits of the brain and not just the moment-to-moment changes. And one of the ways we know when we get all this information, it's a lot of numbers, and what we do is we compare them to a goal group, a very healthy goal group. The goal group we use is all college educated. In fact, half have grad school degrees, which means they're more neotenous. They're more younger minds in a way than the average population. So that is important to be able to take all the numbers and know how meaningful they are. Okay, so the general model that we use for the brain is what I call the two-story living model, where you have the beginning part of our brain is the subcortex, the reptile part. What's the yellow colors down here in this picture below? And when the yellow areas are in charge, as they are in the beginning of life, we think everything's about us. We think we're the center of the cosmos. We're what's called relational thinking. And eventually we start having the neocortex get involved in our ability to understand the world and realize things we call what's all object thinking, where things are independent of us. And that is, we find a balance between relational and independent. That's the healthy point to be. Not everything's independent from us because we alter the environment. That's why things are still relational, but they're not 100% relational as we start off in life thinking they are. So we want to get a balance between the two. And one of the ways we can see the maturation in someone's brain is by the sampling rate. And the sampling rate is easily picked up by the alpha peak. When you have your eyes closed, your brain goes into this idling rate that tells you how often it samples the world, how often it relays information through what's called the Thalamocortical system. And most adults, it's around 10 and a quarter. Some are a little bit faster, some are slower. And there may be some differences in terms of, we've sseen some more ego effects. When people are slower, they seem to be a little bit more egocentric. And above that, what I call the adolescent speeds, 11.5, 12, they seem to be sort of dependent on others, like an adolescent is. And there's been a lot of evidence that our alpha peak rhythm, sometimes it's called the IAF or IAPF for the independent alpha or individual alpha peak frequency, is related to how often you're sampling the world. And one of the, we've known this for over like 70 years, 1949. And there's a recent study looking at flashing and someone detecting 1 or 2 flashes based on how the rate of the flashes. That the flashes are faster than the person's peak, their alpha peak, they see it as 1 flash. And you can see it in the way, if this must be the flash point, they see it or not, or this are the flash points. If not, they only see 1 flash versus 2. So if they sample fast enough, the flashes, you see 2. And that's evidence that the alpha peak is a sampling rate. And as I've shown this one before, the issues when you have slower sampling rates is that you're going to get non-consensual experiences. You're going to perceive things slightly different than other people who are sampling at a different rate from you. And I give a little cartoon analogies or story of someone who has insomnia and how that story is conveyed if you undersample it. Some people pretty much get the correct story. Some people get completely the opposite story. Some people get a different, weirder story. So you want to keep the sample rates the same between the listener and the speaker, between the people you're communicating with, in order to be as accurate as possible in conveying information. There may even be sometimes where the phonemes can be changed, where da may sound like a ba, especially if you're in noisy environments. In pristine, quiet environments, probably not a problem. Have different sampling rates. It's when the environment is chaotic and noisy, then you're trying to communicate, then things are going to go awry when the sampling rates are different from each other. Okay, so we know, as I showed in the previous video, we improve our peak frequency increases almost like a natural phenomenon. We increase about 0.4 hertz a year. We kind of, beginning of life, we sample the world on 3 to 4 times a second, very quickly get up to around 8 frames a second, and then it's about 0.4 on until we hit puberty. And this is actually real data of one of the groups I have of data, and showing there's actually this weird decline. Now, maybe it doesn't happen in all the groups. This is, again, I do think there's a correction goes on right around puberty, where the individual at one time was the center of the cosmos feeling when we were young, with a center, and then realizing, hey, I'm only 0.5, I have to go find my other half. And then I think we stay around this 10 hertz for a long time, or 10 and a quarter, because it's the other people doing it. So we get it's efficient to stay at that rate, to synchronize with others. Just like if you're driving on the highway, it's more efficient to synchronize with the cars around you than to go some absolute speed, faster or slower than the cars around you. Same thing with people communicating. This is just an example of a kid who's 11. You're looking at 3 spectral plots, one age 11, one 13, and one 15. And you can see 11 is a little bit slower. Let's look down here. You can see he's going like maybe like 8 hertz, and here he's getting up to like 10 and a half in the back of the head, and by age 13. Just give you an example how you can see it. Now, there is this evidence that we slow down over time as we hit late middle age and beyond. Now you can see it's very messy data. Some people doing very well. And a lot of EEG is about, I think, just a lot of brain function is use it or lose it. It's like muscle. The more you stay intellectually active, the more you'll be intellectually intact. Here's one of the things we talk about in terms of default network training is we can actually impact the white matter tracks. We can increase the myelination by training between the hubs between them. So we can increase the white matter and also will increase gray matter in unusual ways. And we are by working these systems through operant conditioning. And there is this natural change we see over life where the white matter increases, maximizes, depending on the location, around age 45. Here it's showing around 50, 55, it's maximizing, and then it will decline. And the gray matter, we get more gray matter. I think gray matter is like the computers and the white matter is the wiring, and it's like the more we eventually have more white matter. We have more glial cells, white matter, and other support cells than we have the neurons. That's just natural. We start off with too many neurons, like too many computers on the network, and we strip it down and adapt it to our environment, what's needed. And you can see this loss of gray matter is pretty much all through life going on here. And in most of the neocortex, we do not ever increase our neurons. We're born with our maximum amount. There's a few areas around the smell parts of our brain, the more primitive part of our neocortex, right at the transition between neocortex and the more primitive paleocortex and other areas, that we do have some cells being created. The hippocampus creates cells every day of our life until we pass on. But our neocortex pretty much fixed in almost all the areas, and just there's a decline in area in what you don't need, what you don't use. For instance, the human vocal apparatus, we can make about 60 phonemes approximately, and depending, and our baby is ready to hear, is prepared to hear. An infant's prepared to hear all 60 phonemes, but very quickly, some of those cells that are there to help represent certain phonemes, if the phonemes are not being said by their caretakers, by the people around them, they die off. There's no reason for them to be fed. Those neurons to be fed. They need to be supplied. They just wasted energy if they're not being used. And so, like in English, we eventually drop down to about 45 phonemes. So, and that pruning is a natural process to adapt our brain to the environment around us. All right, now this is actually a study from Gazzeri and Beauregard in 2013 after doing a training in the alpha range, the high end, or the low what's called low beta range, 12 to 15 hertz, F4, P4. They were the two channels trained. And this was in healthy college students. And what they found out is that there was an increase in, very noticeable increase in white matter, like pretty much between the two electrodes, in terms of in the fasciculi, the white matter tracts that were between these two locations, F4 and P4. And you can see that white matter increase. It's a DTI measure here. And there's also gray matter. Now, the gray matter was a little bit more interesting. It had to do with, look like the area, the cell systems that are governing these tracks more than anything else. It did increase the thalamus pretty much right below P4. So thalamus was improved as well. So in a way, if you think of it, with the neurofeedback they're doing, which is very similar to DNT, pushing the person back up this pathway, or accelerating the gray white matter, this pathway. The individuals are in the early 20s pretty much, but that's actually de-aging is going on there in essence in terms of the gray matter.